So today, we're going to talk a little bit about global health and some of the approaches that are being taken here at Scripps. There are many, many global diseases such as malaria and HIV that are particularly problems in the underdeveloped world. And one of the reasons for that is that people can't afford expensive medicines. We're very fortunate in the United States to be able to afford straightforward medicines for these conditions. And so in particular, the Gates Foundation has made it a priority to support development to meet these challenges in global health, which is what I just said. This Caliber Skaggs Institute here at Scripps is really taking the lead on how to do this in the not-for-profit sector. So using the resources of Scripps Mind Trust and some really unique screening resources and chemical libraries that RNAB is going to tell us about, we were able to meet some challenges in this area. So there's a great pipeline for first in class. We're going to probably hear about 3 particular stories from RNAB. And so let me say a little bit about what RNAB is. Okay, so RNAB is a medicinal chemist. Now, so this is a really unique skill set. So we have some brilliant chemists at Scripps that invent new chemistry to try to make incredibly difficult molecules or create new processes, new catalysts to make more complicated molecules. The medicinal chemist has sort of a different task, and that is to take something that comes out of a high throughput screen that might become a drug and turn it into a drug. And it's sort of this balancing act. So almost all drugs bind to a particular target inside a cell. But the thing that comes out of the hit might bind weakly. You need to improve that. But you're also fighting the body. So the body, your liver is saying, what's this? I want to get rid of it. It has to be delivered to the right tissue for the right amount of time to get the therapeutic effect. And so the medicinal chemist's job is to turn this lead molecule into a drug, doing this balancing act by completely engaging with the target, but making it so the body would take it up and it goes to the right place. And so this requires real understanding of pharmacology, biology, structural biology, and of course, organic chemistry. So he's a jack of all trades that really takes the crude output of a high throughput screen and then drives it forward so that it becomes a drug. And so this is an essential arm of the Caliber Skaggs Institute. RNAB is the visionary leader of that arm. And I'm going to let RNAB introduce himself and tell you his story. How did he come to be at Caliber and at Scripps? Now, at the end, I'm going to join you back here up in the front, and we will have some Q&A, have some discussions. I'll have a couple questions, and if you're here in the audience, you can put your hand up, and we'll have runners that bring you a microphone. If you're in the virtual audience, you can type your question at any time into the Q&A. And those will get sent to me on my iPad, and we'll try to get as many of these as we can. And we always run out of time. There's always more questions. And so now, for the first time, we have a mechanism to get RNAB to answer you these questions offline after the talk. So I believe there's going to there's a QR code, but there's going to be a special way for you to ask the questions. More info will be available out there at the reception at the conclusion of the talk. So it's my pleasure to welcome everybody here, but why don't you join me in welcoming RNAB to the stage.
Great. Thanks a lot, Jamie, for that kind introduction. And it's really wonderful to see a lot of familiar faces and some faces that I think have been actually transformational for my career in science, and also some new faces as well. So thankful for you all to be here today. So I'm going to tell you a little bit about, well, I'll skip over that, and I'll skip over that. Scripps is turning 100, okay, great. Okay, good. There's probably enough signs, I think, on Torrey Pines Road to remind us of that. What I wanted to do today was spend a little bit of time, as Jamie pointed out, and thanks for the introduction, a jack of all trades is a good description. I will say, as I go through kind of what my scientific journey has been, I've usually been pretty distracted to do something new after every few years of my scientific career, but I've been fortunate enough to do this business for 20 plus years. And, you know, as thinking about how to go from something that's a scientific concept to actually do something that could actually affect patients, be in a pharmacy, and be addressed for someone's needs, I think is transformational. So the picture on the lower left is obviously a picture of a primary caretaker holding a young child and a person delivering medical practice on the right. I will say it's at least for me and my life partner, our kids were never that excited to receive what appears to be an oral vaccine. Maybe you all have different experiences, but what's critical is that this kind of approach to being able to take care of people, I think at its core, is kind of what inspires me to do the work, not only to mentor and be mentored, but also to eventually have impact and be able to help those. So I will give a little bit more of an example of what actually a medicinal chemist actually does. And it will be somewhat of a pattern recognition, machine learning exercise, hopefully, that you can do without needing GPUs to do that. And then I'll talk a little bit about, as Jamie pointed out, some of the things we've done. I'd say, you know, as Jamie pointed out, taking a molecule and optimizing it to make a drug candidate is really at the core of what a medicinal chemist does. And clearly myself and the colleagues I'm proud to be associated with throughout my career, that's kind of our main purpose. I think also part of what has inspired me, and I'll talk a little bit about that as we move along, is that perhaps there's ways that chemists can actually be involved in the earlier processes, i.e., what molecules are we screening? How do we actually come up with what molecules to screen? How can we actually do that in a way that can actually improve and reduce the amount of time it takes to actually get something into clinical trials and then into patients? So I'd say a large part of my career has been really spent on how to reduce those gaps and how to improve the ability for us to do that effectively. And then I'll also talk about how we are thinking about improving drug delivery. And so by that I mean, you know, I think over the last few years, we've been thinking a lot about how to get drugs in the right format to the right population to be able to really impact them. So as you probably are now all aware, there's a lot of, you know, peptides that people are making for incretins, right? So, you know, I joke often that we're trying to figure out ways to swallow peptides, which is not easy. Peptides don't usually like to get swallowed, and we're injecting small molecules that are actually drugs that can be taken orally. But I think I'll tell a little bit about a story where, and both in malaria and in HIV, where using oral molecules to actually develop injectable drugs where you actually are most efficiently using every single molecule of drug that you've made, which is not cheap, right? That's the reason why we have production capacity issues, environmental impact issues, come from us not taking full advantage of those drugs by just changing kind of our convenience preferences. And then I'll talk about what I think is clearly an urgent need and something that I'm very, very proud that we've undertaken at Caliber Skaggs in the last several years is how do we prepare for pandemics? And I think a nonprofit and an academic institute like Scripps with you know, generous folks who've put money and time and effort into this. Certainly the Skaggs family has been central to that. Their vision has allowed us to really be able to think about not just drugs that have commercial benefit, but most importantly can have benefit in areas that we absolutely need, and that is to protect our community from something that could be even perhaps worse than COVID-19, and I'll talk about that near the end. So Jamie did ask me to tell you a little bit about me. So this is kind of, I actually did this slide in 15 seconds, so apologies for any run-on sentences. But you know, my journey really began in high school, and I'm happy to say there's several high school folks that it's part of a larger high school program at Caliber and at Scripps as an institute to bring in high school students from disadvantaged areas and other areas to be able to do chemistry. I was very fortunate to be able to be in a chemistry lab and start running chemical reactions as a sophomore in high school. It actually is what, you know, allowed me to continue to actually make some money and pay for my car, which obviously back then was the most important thing to me. And so I was inspired by that, and I was inspired by particularly female chemists who were in the lab at Baylor College of Medicine at the time, who were key mentors to me to really be able to show the power and the equality in many ways of doing chemistry, making something new, being inspired to do something new and to create something that can help people. I then went off to undergraduate, and I had the chance to be at Northwestern University with Rick Silverman, who was my undergraduate research advisor, who had invented a very important drug molecule called Lyrica. It was a pain drug that's been used. It was one of Pfizer's largest products. And Rick really kind of inspired me to think about two things. One is that sometimes it's better to be lucky than skilled. He invented a multi-billion-dollar molecule between him and one other scientist at Northwestern. And I think it speaks to kind of why I think Caliber SCAGs is so powerful for me, because I saw at a very early stage in my life the power to do something meaningful in an academic environment without needing necessarily the economies of scale. Not that you don't need economies of scale. I think you certainly do to be able to get to products and to be able to roll out to all the people who need it, but to be inspired and to be creative in doing good science. I like, now I guess they're called gap years, but I took some time off, moved back home, lived with my parents, wanted to feel what it was like to not be a teenager living with my parents again. And I did something outside of chemistry, which was great, because I think it also taught me about the power of using modern computational tools to be able to apply that, and I was fortunate to be able to do that. I moved on to graduate school. After a few years off, I realized I love chemistry more than anything else that I was doing, and had the chance of working with Bob Grubbs at Caltech. With Bob Grubbs, who later ended up winning the Nobel Prize in Chemistry, it was more about learning about how we use new reactions to develop new methods to actually be able to make new molecules. And in fact, there are a half a dozen approved hepatitis C molecules that actually use the chemistry that was developed by Bob Grubbs and fellow coworkers working in the area of olefin metathesis. So while he didn't invent drug molecules, he was generating chemistries to do that. And I think, you know, there are many faculty members, as Jamie pointed out, who address this every day in the chemistry department here at Scripps, which is how do we more effectively make molecules to really impact human health as quickly as possible. And I'm very proud to say that those folks are my colleagues. I left and took a very different route than the traditional academic approach. I was inspired by two places in particular. One was Gilead Sciences under John Martin's leadership, who I was inspired by probably two chemists who've impacted me greatly. One was John, and in the early days, this is 2002, Gilead was a younger company and was doing amazing work in antiviral drug discovery, and I was truly inspired by that. And then I visited across the street to this place called GNF, and I'll tell you a little bit about what we did there with Elizabeth Winsler, Case McNamara, and Adi Nagel, where we actually invented a malaria molecule. I was inspired by Pete's interest in being able to do this, Pete Schultz's interest to be able to do this in a way that we could quickly be able to do what I was discussing earlier, make libraries, make drug molecules, and be able to test them. And from that, I'll talk about a program that was very inspired me and hopefully tells you a little bit about what med chemists do. And then once Caliber started and once Caliber became part of Scripps, it was very clear to me that this was the route I wanted to take, at least for the next journey in my life. So why work on global health? And I think, you know, I've had many discussions with colleagues around, you know, why do this? And, you know, I think we can safely say that if you look at this list, this was updated a couple years ago. So this is the graph on the right is disability adjusted life years. So the amount of time someone is fundamentally affected by disease, that includes mortality, but includes more than just mortality by various diseases. And you can see, you know, diseases like cardiovascular disease that Mike Bolong talked about at the last front row, speak about how can we regenerate heart and repair heart. But then you'll see the next few are diseases that obviously all the ones in red predominantly affect low and middle income countries. But we also know that they affect people here too. COVID-19 certainly didn't leave any country aside in its ability to inflict harm. And so as we're looking through this list, clearly there's a need to be able to not only protect people, but also to grow the next generation of scientists. As I'll talk about later in influenza, I think we got very lucky with COVID-19 that it did not affect as many young people as it did. We know influenza is just the opposite of that. So the ability to really be able to work on these diseases is critical because a disease in any one part of the world can affect anyone else. And so I'm really proud that that's the vision of Caliber SCAGs. And I'm also exceptionally proud that the Gates Foundation, Wellcome Trust, NIH, others have joined in this effort to really be able to use the best minds and the best colleagues and the best environments to be able to really bring together this sort of work. And I think the other key thing that I wanted to mention too, and it really speaks to the heart of Scripps and the vision of the Institute, is to be able to combine really interesting basic chemistry, basic biology with this translational science. And I won't get a chance to talk about it today, but there have been chemistries that have been developed at Scripps, C-terminal decarboxylated borylations, fluorosulfate chemistry that we're now applying to drug molecules that are near the clinic. And I'm very, very proud of working with those folks to be able to do that. But here's a quick story about what drug discovery looks like. I tried to put a lot of work into one slide, and Elizabeth Winsler, who's my colleague at GNF and now is at UCSD, and thankful that she's here today, was really inspirational behind this. So the molecule on the left, the molecule in the middle, and the molecule on the right are all slightly different, but I could argue that they're all like first cousins. Maybe the last two might even be direct siblings. This was a compound series that was generated. The compound on the upper left was generated by a company called Array BioPharma, which sold chemical libraries many years ago, made a library, and GNF purchased this collection. And it was sitting around. There was actually about 7,500 compounds made. There's a publication around this that was made by very simple 3+2 chemistry. It's 3 chemical steps, commercially available starting materials, and they made 7,500 compounds. And they did it because the chemistry was simple, and they could sell the library and make a profit. In fact, Array now is part of Pfizer and has developed multiple drugs that have been approved. But this was kind of their bread and butter about how they kind of started the shop. And from that, it was screened by Elizabeth and her group looking for molecules that inhibit a simple thing, which is malaria parasite in the blood. And she found this molecule. And so with Adi and others, we generated a molecule that's shown in the middle of the slide. That's actually a very interesting molecule. I put a box around that potency number because it's very potent on killing malaria parasites. It has other issues. So then we spent 2 years and 560 molecules to go from the molecule in the middle to the molecule on the right. And if you notice, the only thing that has changed in those 2 molecules has been the location of these 2 methyl groups. So if you take half the analytical methods that chemists use, these 2 molecules are identical. All that's happened is these 2 methyl groups have moved from this carbon to this carbon. And in those 560 molecules we've made, the chemical complexity has gone up significantly. So now you've gone from 3 steps to make those 2 molecules, the middle compound and the left compound, to nearly 12 steps. And Novartis is still using the same route that we developed at GNF and is now in a phase 3 trial. The interesting part is that this molecule is likely, it's in phase 3 right now with approval expected in 2026. This will be the first non-artemisinin-based combination therapy, i.e. a completely new class of molecules that's first in class to treat a vital infection. This drug works in the prophylaxis of malaria and in the treatment of malaria. But it took us a long time, and it took us a lot of molecules to go from the molecule in the middle to the molecule on the right. So the question is, how can we take advantage of modern chemistry and modern biology to kind of improve this process? A Herculean task and really creative work done by many people, but still it took a lot of time. So how can we cut that short? So we started thinking about how we could do that in the context of this concept called drug repurposing, which is building a chemical library and then using that chemical library of known drugs and taking that set of molecules and testing them in every assay that we can. And so I mentioned this a few years ago. We built this collection with foundational involvement from the Gates Foundation. They were very interested in this idea of testing molecules without having to license them all, which you can imagine can be difficult from pharmaceutical companies at times to be able to access, and even more specifically biotechs, and put them in a collection, and share them with everyone. And so this has been, I think, speaks greatly to what the Gates Foundation wanted to do and speaks very much to the ethos of Caliber SCAGs. And so what does that allow us to do? It allows us to take drugs that have been developed in all different disease areas, cancer, autoimmune disease, and others, and apply them to completely new diseases and completely new ways of thinking about how to do this. And the best part of it is that, as Jonathan Tynan and other folks are now doing at the Institute, we make all of this data and all of the compounds available to anyone. The only requirement is that once they use the compounds from us, they publish their data within a year. And you'll be surprised by how many academic institutions, I will not name them, do not like that from an intellectual property perspective, even though they own all the intellectual property of the screen, because they don't want to publish the data. So we, in fact, during COVID-19, not only cut that period from 12 months to a month, we also screened everyone's collections. And I think that really speaks to the mission that we have at Caliber, which I think is really unique and speaks to something that I think we need more of. I mentioned this before, so the collection of Reframe is about 12,000 molecules, and it turns out there's about 18,000 molecules, small molecules that have actually been synthesized and developed and put into clinical trials. So we still have some work to do to make another 6,000 molecules, but you can only buy 6,000 of these 18,000 molecules, which means that when we're testing these compounds, we actually, when people are buying molecules and they're testing them, they're only investigating 1/3 of the available data, right? They are actually missing out on a huge amount of data, and I'll show on the next few slides how important that is to develop new drugs. So, like I said, we have many more compounds to make, 6,000 more compounds to actually procure. The very interesting part is that there's a number here that 92% of these molecules are not even in the world's largest open access chemical databases. So 8% of the molecules in Reframe are not even in databases that anyone else in the world has access to unless you pay a bunch of money to paid for subscription chemical services. So we've been able to procure this data and make it openly available. And what's really exciting to me as we're thinking about using this in the context of machine learning and AI models, we've generated nearly 1 million data points of drug screening information and compound information, i.e. what diseases have these molecules been in, how have they been used, and all of that data is available. In fact, people reach out to us and say, how much do we have to pay for your data? Or how much do we have to pay for your compounds? And we say, you don't have to. You just need to be able to annotate that we've used the data from us, and if you access the compounds, you need to make your compounds publicly available. What's really interesting, as I show on the upper right part of this graph, is that when you ask the simple question of what drugs have been made and put into humans, you get a very different answer depending on who you ask. So I just note here 3 databases that cost several hundreds of thousands of dollars to access. We used them at GNF when I was at Novartis across the street, and you get very different answers. And it's because they're editorializing the data, i.e. they're looking in different databases in different places. This just isn't looking at what FDA has put in. This looks at every molecule that a physician has considered safe enough to put into a patient to treat a disease or to determine its safety. So I think it's really valuable to say this takes a large effort to be able to do it, and a place like Caliber is really a place where we can do that. So Caliber was first developed this 6 years ago. It was a Herculean task in compound management led by Megan Young and led by many other people who I worked with at that time to put this collection together. What do we have from it? And so I wanted to give you all a progress report on kind of what we've done. And I'll talk about some examples. But I can tell you from this screening collection alone, and Mike and others have discussed some specifics, we've actually generated over 14 molecules that have either gone into the clinic or molecules that have made it to the stage where we're determining safety to put into the clinic. And that's 14 molecules in the matter of 6 years screening 12,000 compounds. The productivity of that compared to what we normally do, which is high throughput screening, is you screen 5 million compounds to get to those number of drug molecules. Or in the case of DNA encoded libraries, you're screening 100 billion compounds to get to that many compounds. And so going from that to screening such a small set of 12,000, knowing that there's still 6,000 more that we should actually include in the collection, is really important to lead to this many number of outputs. So I won't go through all of these in detail, but simply to say several hundred screens have been run on the collection across a wide variety of disease areas, mostly in infectious disease, about 75%. And that makes sense given the fact that we really wanted to use this for diseases where there's really no immediate commercial benefit. So things like malaria, things like TB, things like even COVID-19 are listed on this slide. But we've also been able to generate molecules for fibrotic disease, for regenerative disease, and other areas where simply asking the question, can an old drug be applied to a new disease, is meaningful and impactful to really allow the acceleration of discovery of new drugs. So I'll speak to one, vignette and one story around kind of what we've done here. So this is a molecule on the upper left. And so I know this is going to be by far the most chemically structure-heavy slide that I have. So apologize to everyone except for the bona fide chemists out there. This was a screen that we ran. This was done by Melina Bukowski, Tom Rogers, and with an assay developed with Dennis Burton here at Scripps, which actually was the first screen ever to be run on human cells infected with SARS-CoV-2. So this was a screen that was developed in April of 2020. We had run a screen with Sumit Chanda and with folks at Hong Kong University on another cell system, but this was the first human cell system we had run, and we found this molecule on the upper left. This is the molecule that Novartis took into multiple clinical trials for treatment of liver disease, and in fact, while they never got to a successful approved drug candidate, a lot of challenges in the field of finding drugs for liver fibrosis. I think there's now one and a half approvals really in the last 30 years in this space, but more coming ahead, luckily, to incretin peptides and other things. Found an interesting molecule that inhibits the virus. You can see 72 nanomolar shown on the upper left. But the molecule is really developed to go into the liver, stay in the liver, get rapidly metabolized, and then kicked out. And so Elshan Nakat and Gena Gorbovia and multiple other people, I won't go through the names, they're all at the end on the acknowledgement slide, found this molecule to be a very interesting starting point. And I won't walk through all the details of how we get to the molecule, but if you just take the compound on the upper left and compare it to the compound on the upper right, you can actually see there's a lot of conserved pieces there between those two molecules. And in fact, we went from a reframe screen that was done in April of 2020, and within 18 months, we went to doing a phase 1 safety study of this molecule on the right. And the reason why it's important is because the molecule on the right goes from a liver drug, as the upper left, improves its potency, most importantly, improves its pharmacokinetics properties. So as Jamie mentioned, we went from a drug that now doesn't just go to the liver like the compound on the upper left, it now goes in the lung, it goes into every tissue. And so we know that's where SARS-CoV-2 replicates. It replicates everywhere, but especially in the lung. And we went through that optimization process to actually get to a drug candidate that we then licensed with AbbVie. The Gates Foundation funded this work, and we were able to move that together between AbbVie, the Gates Foundation, and Caliber Skaggs. And so I think it's a real example of where we've been able to bring together various different organizations with different areas of interest and areas of expertise and have real impact in terms of clinical development. There's a great story around how we got to this molecule, and I'm happy to tell you more about it. What I'm really excited by is now SARS-CoV-2 protease inhibitors like Paxlovid and other things that are out there is very interesting. There are derivatives of this molecule, CMX990, that's shown on the upper right, that we're now developing within the Institute that actually not only inhibit the protease, which is the viral protein that's made by SARS-CoV-2, it not only works against coronaviruses, but it works against poliovirus, it works against rhinovirus, which is another cause of respiratory infections, that all encode a similar protein. And structural biologists have been talking about for many years that hepatitis C and these viruses and coronaviruses all share this protein that is similar, but no one has found an agent that actually works on more than one type of virus. And so we're really excited with the fact that we have a molecule now that has a broader spectrum of activity, which allows it to be used not only for coronaviruses, but for other viruses, including poliovirus, which we're now working very closely with the Gates Foundation on. And so I think this is a really important thing for us to say, how can we use Reframe? How can we use this collection of 12,000 molecules, take something from it, accelerate the drug development, much faster than the malaria example I gave at the beginning of the talk and develop something that we could actually put into clinical trials. And I'm really proud about myself and most importantly, the colleagues and the collaborators that we've had in this work. So the next part of the talk that I'm going to talk about is another thing that's also, quite near and dear to me. And so this is the concept of using long-acting injectables to reduce drug cost. And I started off by saying reducing drug cost, not only because I think it's critical from a drug perspective, i.e. you are giving a drug to somebody that lasts in their body for several months, which I think from a safety perspective is something that I think hopefully can convince you that that's a useful thing to do, where you have an oral drug that you already know is safe, and now you're just changing the way you're administering the drug. It's no different than what is now happening in the worlds of Novo Nordisk and Eli Lilly, taking oral incretin peptides and making them into oral formulations. So I think one of the things we've been really excited by is how we can not only improve the efficiency of drugs, but we can overall reduce the burden on our public health system to administer these. So now the access to pills every day becomes a lower need. And in many cases where people don't want to take medications because it's inconvenient for them to get the medication and to take it, you know that the drug is actually there. So there's been foundational work in the area of HIV. I could argue not only has HIV done a tremendous amount of work in developing this concept of long-acting injectables, but also, most importantly, is inspired by the use of these types of long-acting injectables as drugs for psychiatric disease. So often in psychiatric disease, especially in schizophrenia, but perhaps in other related conditions, and I think Hugh Rosen talked about this a couple front rows ago, is that the ability to be able to give a drug and know that the patient is actually taking the drug fundamentally changes how effective the drug is. We know that drug adherence, remembering to take a medication every day, or twice a day in some cases, 3 times a day in some cases, can be limiting. So how can we develop a drug that we know we've given to someone and that they have taken it, and hence we know that it will work? And so not only, like I said, has this been used in viral indication as shown on the slide, there are many diseases where we know the efficacy of the drug is affected by skipping doses. And so how can we best do that? So we've developed, and I'll talk a little bit on the next slides about kind of how we've done this. I think one really important thing is that there are mechanisms available through the FDA called 505(b)(2), which allows you to take an oral medication that is already approved because it's safety and efficacy. You can make a long-acting injectable of that drug and allows you to cut the development time by up to 75% in some cases to be able to use that drug as a long-acting injectable. So not only are you utilizing all of the data of that drug, but you're also utilizing the fact that there's mechanisms in place to get those to patients, more readily. And I think especially in the use of psychiatric disease, it's powerful because as new drugs are coming on, people want the long-acting versions of those to be available as quickly as possible because they know that those safer drugs, more efficacious drugs, need to be formulated in that way to really impact patients in an effective way. So really, I'll talk about this for the pharmacologists in the room. I apologize for simplifying pharmacokinetics here to one figure. But to improve adherence, I think one of the things we need to do is to develop infrequent dosing regimens. We think about this often, but if there was a way where we could develop drugs that could just be given once, and I think now we're starting to think about that not only in the context of what I'm going to talk about in the context of malaria, and also what we've done in the context of HIV, but be able to get to molecules that could be given once during an infection, or something like COVID, for example, or pandemic influenza, for example. Could we actually give drugs once when somebody is diagnosed with the disease, and we know that there's enough drug there to be able to do what they do? So this graph just simply shows systemic drug concentration over time. The oral drug is shown as the line that kind of has periodicity in its exposure curve, and you can actually see that there, in most diseases and in most disease mechanisms, there is an effective concentration that a drug needs to be above. That's that dashed green line at the bottom. That can either be a minimal concentration or it could be the area under the curve of an average concentration that needs to be covered. And so when you're taking an oral drug, often you are getting these large spikes in the amount of drug that you have, because a drug only lasts in your GI tract for a certain amount of time before it's excreted. And so if it's absorbed, you'll get this really high concentration, and then the concentration will drop off. And often we know that there are many toxicities, particularly cardiovascular toxicities, that happen once you get to that red dashed line at the top. And so once you get near that level, or the closer you get to that level, the higher risk that you have that you're going to have toxicity from it. So all that a long-acting injectable really does is that it flattens that exposure curve. And so I feel like the term flattening the curve has been somewhat dissuaded from our COVID days. But really, when you're flattening the drug exposure curve, you're now able to stay above that effective concentration for a much longer period of time, because the amount of drug you're giving is the same amount that you're essentially giving in that oral medication, but you're simply allowing it to last for a long period of time, and hence being able to get drug that isn't wasted. So really, you're allowing each molecule as much as possible to be effective over time. And so I think that that's really quite important. And I think that's also important from a drug perspective. Often, we're talking about long-acting injectables where the dose over a certain period of time, say over a month, can be 30, in some cases even 100-fold less than what that person would take over that same period of time if they were to take a pill every day. And so I think that is really critically important. And so the other reason for doing this, most importantly, not only from a drug efficiency perspective, is to actually, this is what many patients actually prefer. So as shown on the upper right slide there, and sorry for the legends being somewhat small, but I hopefully can orient you through this. So the pie chart on the left-hand side is actually a survey of patients in low and middle income countries around what type of formulations they would most prefer. And the most preferred is actually shots and injections. That is that blue part of that chart. And that isn't even under the context of you only have to take this once every few months. This was said as if you had to take a pill every day for a week versus an injection, which one would you prefer? That's the blue part of that graph. And in fact, the other areas are, not surprisingly, the next largest part is oral. And then after that, it drops off to implants and other strategies. And the least preferred method, not surprisingly, in those same groups in low and middle income countries is oral, only because of a factor of convenience and cost. That is what the difference is. And is the ability of being able to know that once you take an injection, that drug is staying there. That drug is going to be there. There's no chance of you excreting it out. The excretion mechanisms via sub-Q injection are fundamentally different and not rapid as they are for oral drugs. To get into more of the details, I'm happy to answer more. We go through a lot of different chemical strategies. I mentioned previously we go through this idea of making prodrugs, so we change the properties of the molecule such that when it gets injected and when it gets released, it actually turns into the active drug. And the FDA and other regulatory bodies allow you to use that mechanism to say what you actually need to then do is just show local safety of that new molecule that you've injected. But since it's immediately converting to a drug that we know the safety and efficacy of, we will accelerate the approval process of that. And so this is a critical technique. We use both the way that we deliver the drugs, the way that we formulate the drugs. And so there's a lot of science and a lot of things that go behind how best to do that. I've been very fortunate to say we've done this with very simple formulations in the examples that we've developed to date. One of these is our work around long-acting atovaquone. This has been done with Anil Gupta and with MMV, who's one of the world's developers of antiparasitics for the treatment of malaria disease infection. There's a molecule that's shown on the left side there that's called atovaquone. That's a part of a molecule called Malarone. It's probably something many of you might have taken for oral prophylaxis of malaria. It's a molecule that's used orally. It's a safe molecule. It was originally developed by GSK back in the 1970s and 80s. We turned that molecule, we added that red R group, which is a simple acetate ester. What that does is it allows us to take the molecule. So we took two things. We took atovaquone and we took this ester and we dosed it into animals. I showed the PK, the pharmacokinetic data there. That dashed line on the bottom around 100 nanomolar is the concentration, or is the effective concentration. That's where you need to be above. And you can see if you take atovaquone and develop a suspension formulation of it and inject it, you also get that high peak. In fact, it's even higher if you give the compound orally. But by making that prodrug and by making a formulation that is a simple formulation that is cheap and safe, and most importantly, a molecule that can survive very, very tough weather, so does not need refrigeration, does not need cold storage, can be produced and saved for many years. This is work that was also done as a collaboration with Janssen Pharmaceuticals, who's developed many anti-schizophrenic drugs as long-acting injectables. You can see that line is now much, much longer. And what's also very interesting is chemically, this acetate ester that we made is actually more shelf stable. So in fact, it's more stable than the parent molecule itself. So not only have we improved the way that the molecule is introduced into the body, we've also made something that doesn't need to be thrown away as quickly because the molecule is fundamentally more chemically stable, and happy to tell you a little bit more about why that's the case. We moved forward with this with Janssen and with MMV, and we're about to enter a phase 1 study in malaria prophylaxis. We've also been able to do this in HIV, so to be inspired by my early interactions with folks there, we've licensed a molecule to them that is a long-acting nucleoside for treatment and also prophylaxis of HIV. Gilead is the world's leader in this space, so we're really delighted to be working with them on it, and that also is now in clinical study, so hopefully we'll learn more about that. I'll finish up with pandemic influenza, only because this is now near and dear to my heart, working with Case McNamara, Sumit Chanda, and others at Scripps and at Caliber around how do we develop new drugs for treatment of an infection that's even worse, perhaps, than COVID. We know from Spanish flu, the early death rates from influenza infection can be as high as 60%. We know that our immunity to seasonal influenza is okay because we have vaccinations that we've done, but we don't have something for new strains of influenza. And so what we've been able to do is take a very broad approach. That's one of the things I love the best about the way we're organized at Caliber is 140 scientists, but we're able to do multiple things in parallel to really get to something that can be unique. And one of those things is actually coming up with a single pill combination of two mechanisms that we already know are approved, Tamiflu, which is off patent, and Baloxavir, which is shown on the right, which is still on patent for several years. So how do we make something that actually doesn't violate people's IP and most importantly can impact patients by being able to combine two mechanisms? We know for flu, and we were very lucky with COVID that we didn't generate Paxlovid-resistant or Molnupiravir-resistant virus. With flu, we already know we're not going to be that lucky. Seasonal flu has become resistant to these two drugs within months, and in the case of the molecule on the right, during the clinical trials. It's also true with the Tovacone, which I showed in the previous molecule. So saying that you have a molecule and saying that you can go home because you're done is simply not enough in the world of pandemic preparedness, infectious disease, drug discovery. And then we have other mechanisms that we've done. I'll say that there's the concept of repurposing, and we've screened drugs for repurposing against flu. There's certainly some mechanisms that look interesting. I'll talk about some other things we've done, for example, looking at anti-inflammatories that can be used for flu, that we now have in mouse models of disease, which I think is really quite exceptional. And then I also talk about this concept of NCs, or new chemical entities, right? Using reframe hits to then optimize and make really interesting molecules like the COVID example and some of the other examples that I showed about in the context of other diseases outside of even infectious disease. So this is work with Chengguang Yu and with Karen Wolf and others at Caliber Skaggs. We've been able to generate novel compounds of not only protease inhibitors, which I talked about on the CMX 990 work, where we found protease inhibitors that work against multiple viruses. We also now found a nucleoside compound, which we know is a mainstay of antiviral drug discovery and therapeutic development, and found one that works against multiple viruses. Works against flu, works against arenaviruses, and it works against multiple other viral families. And so an opportunity for actually finding a molecule that can be used against all types of respiratory viruses, we think is a critical tool for pandemic preparedness. And I'm really excited about not only these molecules on their own, but most importantly, when we combine them. Because when we combine them, we know from the world of HIV and hepatitis C, we know that we reduce the opportunity for drug resistance, and we are allowed to save drugs for a much longer period of time. So how have we kind of tried to reduce that to practice? So, we've been able to take, for example, existing mechanisms of action, like I talked about with Baloxavir and with Tamiflu. We've come up with new molecules that we can combine to be able to do that. We've been able to show in the kind of new mechanisms of action. We're using the reframe collection with Adolfo Garcia Sastre. We're actually testing them directly in the flu flu mouse model, which is really exciting because the flu mouse model is actually quite similar to what human infection looks like, so we found interesting molecules there. We have new mechanisms of viral fusion and one polymerization, new mechanisms to come up with new molecules that can be used as new combinations. And then we also have been able to develop, like I mentioned, protease inhibitors, nucleosides, and other things to be able to combine not only for influenza, but for the next coronavirus, the next rhinovirus, the other viruses of pandemic potential, which I'm really excited about preparing as we do more work here. So as I mentioned, for H5N1, the critical thing is not only do we need to think about it now and be in the lab working on it now, I'm very happy to have a tremendous set of colleagues here at Scripps who've been working on this and the drug development as well with my colleagues at Caliber SCAGs. We've been able to show that that approach can be useful for coronaviruses, developing pan protease inhibitors, for example, polymerase inhibitors, et cetera. And we can also now add more compounds to the Reframe collection with generous support of how we can actually go forward and find new mechanisms, new compounds, new things to try. And very importantly, I think unfortunately, we've kind of gotten sick and tired of COVID-19 in many ways, though it still afflicts many people. We need to think about how we can learn from this and how we can do better. And the way we do that today is by really being able to work on these viruses now, develop drugs that are useful, and be able to move them forward quickly, not only as oral drugs, but also as injectable drugs and inhaled drugs in ways that we can get them to people at the inception of a virus, of a new viral infection. Because if we don't, we're going to get we're going to get all the things from COVID-19 we didn't like. All of the healthcare workers, all of the frontline workers, all of the people who couldn't go to work, couldn't live their daily lives. Because in many ways, this is a bigger risk than even what we faced with COVID-19, especially affecting the younger part of our population, which is really the future of where we go as a community. I did not get a chance to talk about so many other amazing projects that we have here. Not only we talked about some of the clinical things, but some of the other things we're working on with generous support from the Gates Foundation and many others to develop not only drugs for malaria and TB, which we're really excited about, and then also a lot of things that we're doing in other neglected diseases, schistosomiasis, cryptosporidium, et cetera. I think a small group of people working together with a common goal and working together and not being siloed and doing it together and sharing our successes and failures together is something I am probably most touched by. And so with that, many important people to thank. I've left off names, I'm sure. I won't go through this list, but other than to say, not only has it been a fantastic collaboration to work within Caliber SCAGs, work with the larger Scripps community, the larger San Diego community, but also many collaborators throughout the world and generous support from many different folks, not only from the SCAGs family, but from the Gates Foundation, the Scripps family, many people to name, Martin Foundation, et cetera. I'm very thankful and honored to be part of that group and very happy to address any questions you may have. Thank you.
Great. Thank you, Arnab. So we have some visiting dignitaries. The SCAGs Foundation supports research at a number, I think 6 or 7 universities, including Scripps and UCSD, around the western states. And there's an annual symposium where all researchers gather, and it's our turn to host. So the SCAGs Symposium starts tomorrow, and Claudia Luttrell is here. And I would just like to acknowledge her. She's one of the 4 SCAGs siblings. Everyone's here for the SCAG Symposium. So thanks for coming to the front row. So we've got some questions lined up. Please raise your hand, and a runner will find you. So the Reframe library is just, it's such a great idea, and it's proven to be quite effective. And so, one of the things I was wondering about is you started out with a drug that hit something in the liver and ended up with a COVID drug, right? So how many times does that happen? So do you find that it switches target, or do sometimes the antivirals stay as antivirals because they're hitting a similar structure? And what's the
Yeah.
you have enough programs going that you have some information about
Right.
what's happening.
So often I'd say it's about 50-50 in terms of keeping a very similar target and being able to just change the properties of the molecule that you want to use it. So, Mike Bolon gave this example at the last front row of taking something that is a diabetes drug that goes everywhere in the body, and then how do you change it so you can give it so it only sticks out in the lung if you want to regenerate lung cells. Right. So there you're hitting the same target, but you're changing the way that the compound works. In the example that I showed with COVID, we actually are hitting a protease, but a protease that is very different and very far away from the human protease that molecule hits. So there we did have to do more, but you learn a lot. In that particular case, we had other molecules that were developed for more closely related proteases that also hit, but they were not as inspiring to develop the SAR the way that we did, that's shown on that slide. So it's another one of those cases where taking an anti-infective drug and only screening anti-infective drugs to find a new anti-infective is probably not a good strategy.
So you mentioned that people typically screen millions of compounds, tens of millions. So why are people so bad at designing those libraries in the first place?
Yeah, so right. So I think when you design 5 million things, you can be wrong many times. So I think part of what I'm inspired by as a chemist, and I think a lot of it that's happening here at Scripps, right, is when we develop new chemistries and we are actually making more drug-like modifications, can we use those chemistries to make really significant collections? And I think, the example with malaria is one where that's possible, but I think developing new chemistries at the core of being able to come up with really nice libraries. Because at the end, we are only using about, a dozen to 2 dozen types of different chemical reactions. And we know we have many more things in our hands now.
So I have a question from the virtual audience, and this is about the injectable, long-lasting version. So you went by a little fast, and I don't know the answer to this. This is a good question. So did you change the chemical structure of the drug from the long-acting to get the long-acting form? And so you did change.
Yes, so we changed the chemical structure, but we changed it in a way such that when the molecule makes it into systemic circulation, leaves the site where you've injected it, it immediately turns into the active drug.
So it's at the end, it's the same compound.
At the end, it's the same compound.
But it lives longer to get to where it needs to go.
Exactly. And by changing that, and there's many different ways to do that with many different types of drugs, but the key is to be able to do that in a way such that it resides at the injection site as long as possible, and it itself does not have any toxicities to the place where it's introduced.
Right, got it. And another kind of variation on that, but so one of the things that happens with antibiotics or antivirals is you get the emergence of drug resistance. And do you have any information about how that plays into having a long-acting drug versus one that you take it?
Yeah, so there's actually some nuances into the pharmacokinetics, which is kind of that concentration over time curve that I showed. Often you want to use those molecules in combination with other agents. So for example, the thing we're doing now in the HIV space will need to be combined with something as it moves into treatment of HIV. There's also this concept of once you inject into an injection site, you can assume, generally speaking, you assume that it's kind of like a ball. And then over time, that ball is getting smaller, but at a certain point, it gets so small that it rapidly dissolves. So you get this concept, which is called kind of not flip-flop kinetics, for those technical at home, where you actually now go from a point where the drug is there, and then it basically goes away entirely. So the likelihood that you have this very long exposure tail that could be below that effective concentration to generate resistance, that's less likely. So there's a couple different plays in the chemist and formulation playbook to be able to address that.
Any questions here from the audience? We got up at the top.
In layman's terms, what does it really mean to hit, and what are you injecting? What are you injecting this into?
All right.
Are we talking about just conceptual models, mouse models? Are we talking about mice? Are we talking about
We're talking about in humans injecting it either into subcutaneous tissue, which is, into fat, or doing it intramuscularly into the muscle, the way that a general vaccine is developed. And so that can be done in either of those, two places. That's where it's most commonly kind of introduced. And so those tissues have very different properties, so that in many cases changes the way that you do the chemistry such that you can have something last in the muscle for a long period of time or something that lasts in the fat for a long period of time.
Any others from the?
I think there's a follow-up there.
Oh,
Yeah.
sorry, yeah. No, go ahead.
Are there, in other words, do you have volunteers here in San
yes.
Diego
How do
who
we
are
do this?
receiving
Yes.
these?
Yeah.
It usually takes a long time to do any kind of work like that.
Yeah, so we obviously do the animal studies here, and then where the phase 1 studies are usually happening in healthy volunteers, through clinical trial centers. Yeah, not here within Scripps. Yeah.
Okay, we have a question up here.
Hi, amazing talk. Thank you so much. My question is kind of related to the same one about antibacterial and antiviral resistance. What's one of the practical ways that this whole technology, this whole method is being used to predict potentially good antibiotic and good antiviral medications?
Right. So I think one of the things, and I didn't get a chance to go into it in more detail, but as we're finding drugs that, for example, work specifically on bacterial or viral targets, right? We're generating molecules that are hitting a target that the bacteria, which has a pretty large genome, but not as large as a malaria parasite, which is more similar to mammalian genome, or a viral genome, which is exceptionally small. You're talking about working on those targets, potentially getting resistance to those targets, and combining those with agents, either other drug targets or with ways that the virus, for example, enters into a cell or the bacteria gets into a cell. We're working in tuberculosis, for example. One of the really interesting molecules we have that's going to be dosed in humans later this year is actually a molecule that just changes the way that the mycobacteria gets cholesterol, because it needs to build a cell wall and it needs fat, and we change that. So that we're not changing anything in the bacterial targets, but we're changing the way that the bacteria uses getting inside of a host cell. So those are the ways that we've been doing the combinations to think about how to use a viral target or a pathogen target and combine it with the way that the thing actually lives in our body, which it needs things from us to be able to proliferate.
Here's another one in the back. Very good.
Yes, I'm interested in knowing what the efficacy is going to be on the newly developed COVID vaccine, which is supposed to be coming in the next month.
Yeah, so I think one of the things that one of the things that we're doing at Caliber, which is actually not in the majority of the COVID vaccines that have been given, which is one of the directions people are going, is to use a small molecule that actually stimulates the immune system. And so those molecules are called adjuvants. And it was on that last pipeline slide that I went through pretty quickly. But what adjuvants actually do is stimulate the immune system for your body to make antibodies to what that vaccine antigen is encoding. And so it allows you to lower the dose. In fact, in India, 1 billion people were vaccinated with an adjuvant for the first time in a large trial.
For COVID.
For COVID. And what it allows you to do is to be able to stimulate the immune system so you get a better response to the antigen. So you can change the antigen, which is what the next generation COVID vaccines are, but we're now doing studies where we're combining that. We're doing in HIV vaccines, COVID vaccines, other malaria vaccines, et cetera, where you can add that molecule to it. It stimulates the immune system, so you end up using less antigen, which turns out to be the expensive part, and you can get a more robust response. So that's one of the directions that's going in vaccine development and kind of how Caliber is involved in some of those aspects.
So the COVID vaccine is a lipid nanoparticle that encloses the RNA, and so are you just adding the adjuvant to that, and it's
no,
also
it's actually
separate.
So once you do an intramuscular injection, it's like the long-acting injectables. You're formulating it with the vaccine. You're not having to do any. You're just adding two things together. It's like a combination of two drugs, and when you do that, you end up stimulating the immune system.
Right. Great question. We have one over here.
Hey, Arnab, great talk.
Thanks.
I have this very technical question. I saw that you had as a malaria target GTP-CH. Is that GTP cyclohydrolase? And
Yes.
that was one that we proposed back in the very early day. So I want to hear more about this.
Yeah, it's in the early days. I wish we had some structures to show you on things that look good there, but I think I'm going to have to probably tell you that later as we get the screens going and whatnot. I think for that, that's a pretty difficult target, so I think we're going to have to throw a lot of different screening approaches, small molecules, DNA encoded libraries, et cetera. Yeah.
And I saw another, yep, very good.
From somebody who's been tasked with screening lots of these molecules, people will come to me with libraries of rationally designed chemicals, things that they've put a lot of care, a lot of time and energy into exploring this XYZ space, this chemical space that you have access to. And then they just say, hey, can you screen these? Hey, can you test these molecules for me? And there's never like a sit down between these scientists, and me in particular, having sort of rational direction on how to screen these molecules. And it seems, especially from the Scripps point of view, excuse me, there's a lot of time and energy that goes into creating new chemicals, creating new ways to generate new chemicals, creating new ways to make these drugs, but not new ways to necessarily screen them, or new buffers to put them in, new compounds to put them in, new ways to test them in the biological space that may allow them to be interrogated in these ways that switch them from one molecule to another, or do these special things that we haven't been able to see. And I guess my question's a little off the beaten path, but do you have any recommendations for people at Scripps to maybe talk to on how to better explore these chemical spaces?
Yeah, no, so I think one thing, and I didn't get a chance to talk about that today, because I think with what we're doing, so for example, we're coming up with new and better ways to actually interrogate the chemical space that we get from a screen, such that we can actually more efficiently take screening data, no matter what format it's in, including new formats, and come up with ways to better optimize those molecules such that we can generate really meaningful hits. So for example, if you're doing an SPR screen, or if you're doing a DEL screen, or you're doing a cell-based assay, how do we take all of that data together to really figure out what chemicals are really interesting to design and to make? And so I think part of it does require some new engineering techniques and methods to do new screens. But I think a bigger part of it for me personally is how do we take all that screening data, and Reframe's one such example, and interrogate better ways to do chemistry so we can more quickly make drug molecules.
All right, listen, we're past the hour, so I want to wrap it up. We have one question, one last that I know you know the answer to, which is, so if you're a high school student,
Yes.
and you want to pursue a career in medicinal chemistry,
Yes.
how do you get your start?
Come and talk to me.
So just some housekeeping before we go. That there is a way to continue the discussion. If you have a question that you couldn't ask, or that RNAP has a mechanism, get the QR code here. It's out there too, and we'll get the answers, and we'll do a little follow-up video with RNAB telling you the answers to these questions. The last thing I want to do is remind you that the next front row is September 18th, and it's Artem Paidapoutian, a wonderful speaker who tells about sensory neurobiology, and he's a terrific communicator, and it's something that everybody has experienced. He's talking about menthol and minty fresh and heat transfer and sensing pressure. It's a remarkable lecture. I encourage you to attend, and if you do come, you'll find me in the front row.